Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
What keeps bones able to remodel themselves and stay healthy? A team from Charité Berlin has discovered clues to the key function of non-collagen protein compounds and how they help bone cells react to external load. The scientists used fish models to examine bone samples with and without bone cells to elucidate differences in microstructures and the incorporation of water. Using 3D neutron tomography at the Berlin research reactor BER II, they succeeded for the first time in precisely measuring the water diffusion across bone material, with a surprising result. Around 500 million years ago, early vertebrates in the seas became fish, adopting an inner skeleton and a flexible spine based on a nanocomposite of fibers and mineral, known as bone material. This invention of evolution was so successful that the basic structure was also adopted for later vertebrates that lived on land. However, while the bones of all terrestrial vertebrates are basically equipped with bone cells, osteocytes, certain fish species continue to evolve and finally manage to create a more energy-efficient material, bone-lacking bone cells, found today for example in fish such as salmon, medaka or till. Samples with and without bone cells. We asked ourselves how bone samples with and without bone cells actually differ in their microstructures and properties, says Professor Paul Zaslansky, who heads a research group at Charité Berlin and specializes in mineralized biomaterials including teeth and bone. Together with PhD student Andreia Silvera and international partners, they have now compared bone samples from zebrafish and medaka. Both fish species are of similar size and live in similar conditions, so their skeletons must withstand similar stresses. However, while zebrafish have bone cells, the skeleton of medaka do not. The background to the question is that the function of bone cells in bone and how they change with age is of great interest to the aging population, Silvera explains. Bone cells can respond to physical stress by sending biochemical signals that led to the formation or resorption of bone tissue, adapting to load. But with age or in diseases such as osteoporosis, this mechanism no longer seems to work. With our basic research, we want to find out how bones with and without bone cells differ and cope with the challenges of external stress, Zaslansky says. Strength and elasticity. Bones have a complex structure. They comprise nanofibers of collagen and nanoparticles of mineral but also other minor ingredients. Certain protein compounds, so-called proteoglycans, PGs, are embedded in a tissue of collagen fibers and nanocrystals and play important roles in tissue formation and maintenance. PGs may be compared to salt in the soup. Too little or too much of it is not good, Zaslansky says. The PGs can retain water, and there are plenty of PGs in healthy cartilage, making it as elastic as a sponge. Together, these components form an extracellular matrix, ECM, a 3D structure that provides strength and elasticity, ensuring function for many years. In bones, an open network, lacunar channel network or LCN, of channels and pores with diameters ranging from a few hundred nanometers to micrometers is created in this 3D structure. This LCN hosts the bone osteocytes, cells that sense load and orchestrate bone remodeling. In the LCN and within the nanocomposite, bone contains up to 20% of its volume in water, with many functions including toughening and adaptation to mechanical stress. Neutron tomography at BER II to determine the amount of incorporated water. The researchers first immersed bone samples in water and transilluminated them with neutrons, provided by the Berlin experimental reactor BER II** at HZB, followed by saturation in deuterated heavy water, D2O. 3D data was collected again, and the difference between the two bone states allowed the team to determine for each spine vertebrae the precise amount of water displaced by diffusion of the D2O. In addition, we examined sections of the bone samples, analyzed them by electron microscopy and micro-CT, and we also determined the Pg concentration with Raman spectroscopy, Silvera explains. Surprising results. Pgs make the difference. Until now, it was assumed that both bone types contain similar amounts of water and had very similar composition and properties. In fact, however, the neutron examination showed that the bone material of zebrafish releases half as much water as that of medaka. This is all the more surprising because these bones have a very similar microstructure of mineralized collagen fibers, but zebrafish also contain large cell spaces within the LCN. My first reaction was, this must be wrong. So we checked everything thoroughly and realized it's really revolutionary, recalls Zaslansky. The only explanation for the difference is that the bone matrices of the two species differ in a fundamental compositional component that affects water permeability. And here, both histological studies and Raman spectroscopy show, it's the small but important contribution of Pgs. The medaka samples contain far less Pg than the zebrafish samples. This is a new finding. Although both fish cope with similar stresses, their bone materials do not have the same water permeability properties, says Silvera. New insights for medicine? We hope these results will help us better understand bone diseases as well, Zaslansky says. Why are some bones better at responding to stress than others? What happens when bones age? Could it be that they lose Pgs, and become less watertight? Perhaps aging or pathology such as osteoporosis changes the bone that surrounds bone cells, which makes it difficult to remodel and form bone tissue that works correctly? After 40 years of successful neutron research in Berlin I. The research reactor BER II was finally shut down at the end of 2019. The shutdown was decided by the HZB Supervisory Board in summer 2013. After 40 years of successful neutron research in Berlin I. The research reactor BER II was finally shut down at the end of 2019. But until now, teams are working on the experimental data to gain new insights into materials of all kinds. Andreia Silvera, Nikolai Kardylov, Henning Marcotter, Elena Longo, Imke Greving, Peter Lasch, Ron Shahar, Paul Zaslansky. Water flow through bone. Neutron tomography reveals differences in water permeability between osteocytic and anasteocytic bone material. Materials Design. 2022. 224 to 111275 doi. 10. 1016, J. Mattis. 2022. 111275 Helmholtz Zentrum Berlin für Materialien und Energie. Neutron experiments reveal what maintains bones in good function. Science Daily. Science Daily, the 21st of December 2022. ift.tt/G576 Fuck. Helmholtz Zentrum Berlin für Materialien und Energie. 2022, December 21st. Neutron experiments reveal what maintains bones in good function. Science Daily. Retrieved December 21, 2022 from ift.tt/G576 Fuck Helmholtz Zentrum Berlin für Materialien und Energie. Neutron experiments reveal what maintains bones in good function. Science Daily. https://ift. tt. g 576 fuck. Accessed December 21, 2022. Thanks for watching. Please subscribe my channel for more videos.